In our last class, we talked about how to analyze proteins. Actually, primarily, we talked about how to separate proteins. Okay, so again, like what we mentioned, if you want to study a particular protein, the first thing you have to do really is to purify the protein. Okay, if you want to get the structure of that protein, you need to purify it. If you want to look at that protein biochemical properties, you need to purify. Okay, so today we're gonna take a look at how to analyze proteins. What are some methods? Okay, like what can you analyze? Of course, we can analyze things like approximate molecular weight using SDS page. We can look at the protein's PI, isoelectric point, using isoelectric focusing. Okay, we can do that. We talked about that. So today we're going to talk about Edman degradation, which is sort of like a amino acid sequencing technique. We're going to look at mass spec, okay, and we're going to show you a video for like 15 minutes, and then we're going to talk about how to synthesize peptides, oligopeptides, short peptides. We can talk a little bit about that. And then lastly, we'll get into X-ray crystallography, okay, how to get the structure of a protein. So let me bring back to this slide where we talked about purification, right? We talked about purification in our last lecture. Now, we're going to look at some information about proteins. Okay, first thing is we're going to look at how do you determine the amino acid composition. And it's actually quite simple. All we have to do is take your protein, hydrolyze it, okay, break it apart, and then you look at what are the amino acids inside. Okay, that's one thing. And then we're going to be looking at protein sequencing, and then of course mass spec and protein structures. All right. Amino acid composition, that's actually quite easy. You can do this thing where you analyze the amino acids, okay, and that will give you approximately what is the composition of the protein, okay. And the steps involved are, number one, you first do hydrolysis, okay. Essentially, you take a very concentrated acid, you add it to your protein, you let it incubate for a little while, okay, and then your strong acid will then break your proteins into their corresponding amino acids, okay? And then what you do is you would separate the amino acids, so you do a separation of the amino acid in the mixture by some kind of chromatography. And in most cases, we're going to use anionic, this is ion exchange chromatography. That's what this IEC stands for, okay? And then after you separate them using chromatography, and then you can do some quantitation, okay? You can quantify the amount of a particular amino acid that's in your sample. Okay, so let's say you have a protein of 300 amino acid residues. You hydrolyze them, run them on a chromatography, and then you can see the relative abundance of each amino acid. Okay, now using an ionic ion exchange chromatography, the acidic amino acid elutes first. Okay, because if it's amino acids that are charged, okay, they're going to be the ones that are acidic comes out first, followed by the hydrophobic ones, and then followed by the aromatic ones, and then lastly, you have the basic residues that come out least, okay, because you have some, if the column is minus charged, then your basic residues are going to be positively charged, and then they're going to interact with the column, okay, so that they come out at the very end. Now, amino acid analysis has some limitations. Okay, for example, prolines, okay, react slowly or not at all. Okay, so prolines may be harder to see in this case. As you can see, how proline has a very small signal. Now, the reason why it has a very small signal, okay, is because typically these amino acids, in order for them to be detected by your, in this case is HPLC, okay, is that they typically have to react with another chemical compound, okay, such that your amino acids are functionalized and has an absorbance. And in this case, your proline does not really react with that chemical very well. so that proline has a very small absorbance. Okay, so proline is not great. Right? Another limitation is that the amides of your asparagine and glutamine are also hydrolyzed. As a result, you'll get glutamic acid and aspartic acid. Okay? Because using strong acid, what this process does is it hydrolyzes your peptide bond, right? Your amide bond. So as a result, you also hydrolyze the amide bond found on your asparagine and glutamine. Okay, so unfortunately, you also do that, and as a result, what you see is ammonia. Okay, you will get free ammonia coming out. Also, you have another problem, that is that some amino acids are destroyed. For example, the tryptophan, serine, threonine, and tyrosine. Okay, these are destroyed upon strong acid incubation. Okay, but of course, you can tune the amount of acid and the time of incubation, and then that will allow you to see, for example, threonine and serine. But if you react it for a long period of time, then you damage the amino acids. All right. So that was to get the sort of the composition of amino acids. And for the most part, you do have a little bit of limitations, but you can see a great deal of your amino acid composition. Right? So that's good. You can still see about, let's say, your protein is 5% cysteine or 5% something. Right? Now, once you do that, we want to know a little bit about the protein's sequence. Okay, just knowing the composition is not enough. I want to know the sequence of the protein. Okay, what is the protein made out of, and in what order? Okay, so the first thing I want to introduce to you guys is the Sayer's reagent, and this is the way that Sayer's reagent works. You start with a polypeptide, and you use the Sayer's reagent called FDMB. That looks like this. Essentially, it has two nitro groups on a phenol. Okay. Then essentially what happens is you react the Sayer's reagent with your polypeptide, and it specifically targets the N-terminus. Okay. So it targets the N-terminus. And then the N-terminus will be modified by your Sayer's reagent. Cool? With me so far? Then you hydrolyze your amino acid, okay? And then you break the amino acid in parts. Now, all the other amino acids are just free, normal amino acids, but the first amino acid which you labeled, essentially, right? The first amino acid on the N-terminus that you modified will be different. Now, you then separate all the mixtures using HPLC. Whichever that has this particular group, is on the N-terminus. Okay? So you can do that. So using this method, I can know exactly which amino acid is on my N-terminus. Okay? Because you look for this particular group, and then you look at what is the R group of that particular amino acid, then you know who this guy is, right? And then using this reagent coupled to partial hydrolysis, Sayer, the same guy that did the Sayer sequencing, determined the sequence of insulin, okay, about 60-something years ago. And it turns out insulin was the first protein to be sequenced, okay, and he actually won the Nobel Prize for this process. So the question is, how do you get this particular molecule onto your polypeptide, right? Okay, this molecule reacts with amine. Okay, it reacts with an amine group. Okay, and you have a free amine on your N-terminus of your polypeptide. Okay, so you add them together, react. Okay, and it's very specific. All right. Now, that's great, but one of the limitations would be you only get to know the N-terminus of your protein. Okay, so what you have to do is you have to first do some fragmentation, and then you look at the N-terminus of each fragment to determine it. It's great. It was like the first protein sequencing thing, but it's not ideal. Okay, so nowadays we actually use a different process. Okay, this is called the Edman degradation. Okay, the Edman degradation is slightly different, okay, but using pretty much the same concept, okay? Now, what Edman degradation does is it modifies and cleaves the N-terminus residue, okay? So in our previous method, the Sanger's reagent, it only modifies. It doesn't cleave, right? So the cleaving is done by nasty hydrolysis, which also cleaves everything else, okay? In this case, the Edman degradation, what it does is it modifies the N-terminus and it also cleaves the N-terminus number 1 residue. That's good, okay? So let's take a look at the chemistry a little bit. So what you do is you use this thing called the phenyl isothiocyanate, okay? So in short, we call it PITC, okay? And what happens is if you incubate your PITC with your amino acid, your N-terminus amino group right here will do an attack on PITC, okay, such that you have a PTC derivative of the peptide. Okay, so you make a covalent bond between the PITC with your protein, okay, your polypeptide, at the N-terminus. Okay, so you create this structure. Now, your reagent here can then react with your carbonyl, the first amino acid, right? So then, after it reacts, it cleaves off this amide bond. So you're going to chop off that bond, right? So this is a very simple substitution, okay? So then you have this part of the molecule isolated becomes this, okay? And then the rest of your polypeptide is left by itself, all right? So here, what then you do is you would take out this particular, so you modify it a little bit, and then in the end you get this phenolthiohydration derivative. And then from this molecule, you can analyze it, compare it to a standard of an amino acid. So this is, the blue is the amino acid. And you can create a sort of standard. Okay, so you take this phenyl isothiocyanate, you react to the 20 different amino acids, you will have 20 different of this kind of molecule, and then you compare your sample with the 20 different standards that you have, and then you will know what is the identity of your amino acid. 好，这个 overall process 应该还 OK 嘛，对不对？这个 overall process 呢，你就是有一个这个 organic molecule， 这个 organic molecule 可以跟你的 N-terminus 的第一个 amino acid 做 reaction，OK？ 然后它的 reaction 很特别，就是它会同时把你第一个 amino acid 切下来 ，OK？ 然后让你的第一个 amino acid 呢跟这个 organic molecule 接在一起 ，OK？ 那所以这一系列的反应 react 完之后呢，你就会得到这样子的 molecule， 对不对？ And then 你剩下的 polypeptide 就 free 了 ，OK？ 那我们要怎么知道 based on 我们已录下来的这个 modified 的 amino acid， 我们要怎么知道它是谁？很简单，你今天你只要拿同样的这个 phenyl isothiocyanate，OK？ 拿它去跟你20个不同的 amino acid 先去 react， 那你是不是就有20个不同的这种 modified 的 amino acid， 对不对？那你就知道，比如说你用 HPLC 去分它们的时候，你就知道它们是什么时候出来 ，OK？ 然后呢，你做这个真正的 Edman degradation 的时候，你每一个 isolate 出来的，你都会有一个这样子的 chemical， 然后你跑，比如说 HPLC， 或是你做 some kind of detection， 你就知道说 OK， 这个 according to 这个 R group 是谁，你就会知道说它原本是什么 amino acid。Make sense 吗？可以哈。Right? Now, the beauty of Edman degradation is that it releases your amino acid -1, okay? Such that this process can be repeated. Okay, so what you can do is then you can bring this guy back up on top, and then you can do this process again. Okay? So what happens is that the remaining polypeptide can undergo this process again to release the second residue, and then the third residue, and then the fourth residue. Okay? Every time you go through this process, you eliminate one residue. Okay? Now, this process, okay, so it's a commercialized process. If you have a protein that you're studying, you want to know the N-terminus sequence, okay, you can actually send your protein to a company and then they can do this for you. Right? So currently, we can synthesize, we can sequence about 50 residues. On your slide, it may say more than, cross that out. Usually we sequence about 50 residues from the N-terminus, okay, in a polypeptide, right? So currently there is still a limitation about 50, roughly speaking, 50 residues, okay? You can't sequence the entire protein, and you can think about why, okay? Also, in order to use this Edman degradation, you need to have some prerequisites. Number one, your N-terminus must not be blocked. Sometimes if your protein undergo a post-translational modification, then your N-terminus may be blocked. Okay, so in our 2 lectures ago, we talked about some post-translational modifications, right? If your protein's N-terminus is post-translationally modified, then Edman degradation will not work because you don't have the N-terminus amino group. Okay, then it will not work. In addition, your cysteine also has to be modified because your reagent reacts with thiols. Okay? So to do that, what you do is you have to essentially throw a protection group on your thiol, on your cysteine. Okay? And essentially we use this iodoacetate, which will modify your thiol such that you add onto it essentially a acetate group. Okay? Sort of, this is carboxylate and then a methyl group, so you add 2 carbon and then the terminal is an acid. All right, so after you do that, then this guy will not be touched by your organic chemical in this Edman degradation. Now, anyone want to ask me, how come? Well, actually, I should ask you guys this question. How come? Why do you think we can only get about 50 residues? If this entire process can go on and on and on, why do you think we can only sequence 50 residues? Why can't we sequence the entire protein? And that's a question that you guys should think about. Essentially, it has to do with the efficiency of each step, right? Okay? Now, if you assume a 100% efficiency of each step, then it's okay. Okay? Then it's okay. However, in any chemical reaction, we know that 100% efficiency doesn't really happen. You can get to like 99%, you can get to 99.9%, but 100% is usually hard to do, right? So as a result, you do have an inefficiency in your chemical reaction. So let's say the first one, okay, the first time we do this process, you are left with 1% of the original base, okay, the original protein. 99% has been chopped off. Okay, let's say we have that. So let's say every time your reaction is 99% efficient. Okay, so gradually what you notice is that the original protein will accumulate, or not really accumulate, but gradually by the time you do, let's say, the 50th time of this reaction, you'll have a mixture of polypeptides. Okay, because every time you have, let's say, 1% of the original polypeptide. The second time you do it, you have another 1% of the original peptide. Gradually, the signal, okay, that you want is diluted by the noise, okay, because of the chemical reaction not being 100%. Okay? So gradually, you start to have noise. And because the noise becomes larger than what you want, so after a while, you can't actually interpret what is the next amino acid to be cleaved. Make sense? Okay? Now, let's say you first start with an amino acid that is 300 amino acid residues, right? You run through the first run, and you're supposed to be left with a polypeptide that's 299 residues left, right? However, if the entire process is not 100%, you'll get in your mixture 99% of the 299 residue and then 1% of the 300 residue polypeptide, right? So you imagine for every step you have this inefficiency, and then gradually by the 50th time you do this, you have a lot of different peaks that can come out, right? Because this process essentially cuts the first amino acid out, right? But maybe by 50th cycle, you have different polypeptides, right? And then the signal essentially gets diluted. Right, so this is an automated process. Not too bad, okay? So knowing that, what can we do? By the way, anyone want to ask me how come they don't just continue to react? Like, for example, the first time you do it, why does it only react with the first base? How come you don't chop it off and then you still have the reagent, and then the reagent immediately reacts with the second base? You think about that. If you can't figure it out, you can ask me later, okay? Think about that question. Okay? 你们知道我在问你什么？就是说，你今天有300个 amino acid residue， 对不对？你放在这个 vessel 里面，然后你加了这个 reagent，OK？ 那第一个 amino acid react 了 ，OK？ 然后就放出来了嘛。那为什么你的剩下的 reagent 不会直接去跟你第二个 amino acid 直接 react？ 因为如果会的话，那你就头痛啦，对不对？因为你的 sample 就会 immediately 出现好多个不同的 peak， 这样。Think about that, okay? You can actually see the slide, and it actually tells you a little bit of something, okay? All right, so turns out, okay, we don't have to really sequence the entire protein. All you have to do is fragment the protein first, okay? And fragmentation, we talked about 2 lectures ago about how you can use some reagents or proteins to break your peptide bonds. And these are some common ones that we usually use. We have the chymotrypsin, the trypsin, and cyanogen bromide. The reason why we use these selected proteins like proteases or molecules to degrade your protein is because they're quite specific. Okay, so if today I have a polypeptide and I applied trypsin, okay, and I know trypsin will cut at lysine and arginine, okay? And then immediately I see 1 fragment, 2 fragment, 3 fragment, 4 fragment, 5 fragment. That immediately tells you that you have how many lysines or arginines, right? It already tells you some information based on that, because you know that trypsin is specific for lysine and arginine. So at least you know the location of maybe arginine or lysine, right? So you at least know that you have 4 lysine or arginine. So it's very useful to use these specific ways of chopping up proteins instead of a random way, okay? Now, in order to do your sequencing, you also have to remove disulfide bonds, because disulfide bonds will link your proteins together. So then it will not, if you follow the last slide about Edman degradation, then it will give you problems. Okay, so we have to remove disulfide bonds, right, to separate them and then let them become essentially a linear polypeptide such that you can do Edman degradation 1 residue at a time. So to remove disulfide bonds, what you can do is you can use porphoric acid, which will essentially oxidize further. Okay, you'll oxidize the sulfur into sulfates, so you're oxidizing it. Additionally, you can also do reduction. Okay, so this is oxidation. Then we can also use a reducing agent where we can add beta-mercaptoethanol. Okay, so beta-mercaptoethanol is pretty much just like ethanol, but instead of an alcohol, it's a thiol, right? And it's a reducing agent. So you add beta-mercaptoethanol, then what you get is the thiol that you're supposed to find on your cysteine. Okay? So you can then sort of cut off the disulfide bonds using either performic acid or beta-mercaptoethanol. Cool? And then you can use your sequencing, right? These are some of the other ways that we can do fragmentation, like you separate them into chemical cleavage or enzymatic cleavage. Cyanogen bromide cuts methionine. Iodosobenzoates cuts tryptophan. Okay, and also which side they cut, also pay attention to that. Okay, and then enzymatic ones, we also have trypsin, chymotrypsin. These guys are used very frequently. Sometimes you also use thrombin, which cuts at the carboxyl site of arginine, and so on and so forth. Right? So this chart you should be familiar with. Okay, I will throw in a question on your exam, essentially asking you, let's say I have a peptide or a short polypeptide. I cut it with blah blah blah. I get how many different fragments, and then I want you to piece them together. Okay, so that's something that you should be able to do. Now, in order to do that, you will have to know what chemical or what protease cuts where. All right, so these are just some ways that we can cut proteins. All right, so that's what we have on protein sequencing. Now, of course, nowadays, you don't really have to rely so much on Edman degradation because we have very powerful computational tools. And computational tools are oftentimes used with mass spectrometer, okay? Because we know the mass of each amino acid, okay? And then we can sort of look at the mass of your polypeptide, and then you can calculate based on bioinformatics what the protein might be, right? So mass spec is used to provide an accurate mass measurement compared to SDS page, which is like really rough, okay? Mass spec can actually give you a number, okay? SDS page cannot. And for analysis of biomacromolecules such as proteins, we need to use a soft ionization, right? Have you guys taken classes on instrumental analysis? So you guys know about ionization methods? Right? For example, in GC, gas chromatography, you need to have an ionization of your molecule, and then when it hits the detector, you get a signal, that kind of stuff. Right? So ionization process, essentially it just means that we ionize your molecule and such that the molecule becomes charged, and then when it passes through a electromagnetic field, it's going to have a mass to charge ratio. That is how you know the particular mass of your compound. Okay, so if you guys are not familiar with mass spec, you can come to me and I'll give you a very brief introduction to mass spec. But you know, essentially, what happens is in mass spec, what you want to do is you want to put a charge on your molecule such that the molecule would have a mass to charge ratio. Okay, and you can then separate molecules based on this mass to charge ratio. Okay, now for proteins we have to use soft ionization. Why? Because extreme ionization methods, okay, will actually degrade your protein. Right? You'll destroy your protein. So if it's destroyed, then the mass is not meaningful. Okay, so we have two very common soft ionization methods. There are actually more, but I'm just going to introduce you to two. One of them is electrospray ionization, which essentially you throw charged particles, okay, in a way, like helium, charge it, you throw them onto your molecule, and then your molecule becomes charged, and then you let them pass through your mass analyzer, and then depending on when it hits the detector, you will get a particular mass to charge ratio. Then you can sort of look at what that thing might be. Now, the limitation of ESI is that you still cannot process very, very large sample. So ESI is used for a lot of times not very small molecule, but medium-sized molecule. So that's what ESI is used for. And ESI is pretty relatively inexpensive. It's not very expensive. So a lot of labs will have this instrument. Now, for most mass spec purposes, for especially measuring proteins, most people will actually use MALDI, okay, which stands for Matrix Assisted Laser Absorption Ionization. What it is essentially you put your protein on a matrix, and in this case, matrix is a chemical that absorbs light, okay? And this chemical that absorbs light will absorb the light from the laser, okay? And any chemical that absorbs light, essentially, you're going to increase its energy. So this chemical will have a large energy, and then essentially it will pass some energy onto your protein and make your protein charged. Okay, so it actually charges your protein and essentially throw your protein into the mass analyzer, and then your protein is charged, go through the mass analyzer, and then it hits the detector at different times, and then you will be able to get an accurate measurement of your mass to charge ratio. Okay? Now, what we can then do is use a tandem mass spec. So this is mass spec on top of a mass spec. Okay? So what it looks like is this. You have a electrospray ionization. In this case, we use electrospray because we're not worried about degrading your protein. We actually want to sort of separate your protein and then fragment it. Okay? So you have two mass spectrometer. The first one, right, after you electrospray your sample, the proteins are then fragmented, okay? And you have a lot of different fragment that comes out. And the first mass spec sort of serves as a separation filter, okay? You filter and you filter different sizes. So your mass spec can select a particular ion. In this case, we call it the parent ion. And then you let the parent ion go into a collision cell, and this collision cell will impact your ion with a high amount of energy, okay, usually with high energy argon gas, and then it separates your fragment further, okay, breaks it apart, such that you break it into small fragments, and some of these small fragments are amino acids, okay? So once you break it into these small fragments, what you get is a collection of these fragments will be the breakage of peptide bonds. So you know that some of these fragments that you get from the collision cell will be subsets of small peptides from the parent ion. And since the mass of the 20 amino acids are known, okay, so essentially you can very easily calculate what the short polypeptides would be. Okay, for example, if it's like glycine plus alanine, you know a particular mass. If that mass number matches the mass that you observed, then you probably know that it's going to be a combination of alanine and glycine. Okay? So given that, you feed it back into your computer, you search it into a database, then you can identify what protein this might be. Okay? And it will actually spit out a percentage of what they think this is. Use that, then you know in your sample, this is the type of protein you have. Okay? And of course, you can do this on a large omics scale. So you do a lot of sample, you process crude sample, you don't even need to purify your sample at all. Right? You just take a mixture, feed it into the mass spec, and then you get a lot of results. And then you use bioinformatics to organize them, okay, to then calculate what the samples are, and what the proteins are, and then you can draw some linkage between this disease and these proteins, okay? So this is currently what people do in these things, okay? All right, my next topic is about protein structures, right? So we talked about how to purify protein, how to separate proteins, how to look at protein compositions, how to characterize the protein based on molecular weights, etc. Right? So now we're going to look at the structure of the protein. Okay? So I'm not an expert at this. If you want to find an expert on this, you can go and talk to Professor Xiao Yuan in our department. Right? He does protein X-ray crystallography. He's really good at it, so you can get some insights from him if you're interested, right? But very briefly, we can use protein structures for what? Do you guys know? We can use it for a lot of different purposes. We can do drug design, okay? If you know that a particular receptor is responsible for a particular disease or a particular protein is responsible for a particular disease, you can design small molecules to fit that protein. How do you know what kind of small molecule will fit that protein? You can first crystallize the protein, get the protein's structure, and then you use bioinformatics methods or tools to help you design such a molecule. One application right there. Another application, in my lab, we're interested in designing synthetic metabolic pathways, but synthetic metabolic pathways don't exist because we're asking cells to do a synthetic function. In this case, I can use structure of the protein to help me design a new protein. That could be a good application there, right? So having the protein's structure can help you do a lot of different things, okay? Now, protein structures can be determined using X-ray crystallography. It can be determined using NMR. It can be determined using cryo-electron microscopy. Right? Now, we're only going to talk about X-ray crystallography because X-ray crystallography has a long history and it can give you very, very good resolution. NMR is currently used to handle smaller size things like RNA structures, smaller protein structures. Cryo-EM, okay, cryo-EM, do you guys know what cryo means? It means really cold, right? Low temperature. So cryo-EM is essentially you use EM to look at the protein's structure, okay? It's great, very easy, but resolution not as good as X-ray, okay? So we're going to primarily talk about X-ray crystallography. Now, X-ray crystallography provides the finest visualization of protein structure currently available. Okay? So it gives you a very good resolution, right? And this technique can reveal the precise three-dimensional position of most atoms in a protein molecule, right? And the use of X-ray provides the best resolution because the wavelength of X-rays is about the same length as that of a covalent bond commonly observed in biochemicals. Okay? Now, the way it works is like this. Step one, you have to first get a crystal of your protein. Okay, so proteins have to be crystallized. Now, this example is myoglobin. We'll actually talk about myoglobin in a couple of weeks. Myoglobin is the protein that sort of binds oxygen in your muscle cells. Okay? Now, first, you are going to add some kind of a salt or some kind of a salt solution, and you let the salt solution and your protein incubate, okay, for a long time, okay, and then gradually your proteins will precipitate, okay, it will actually aggregate together and then form a crystal structure, okay. So you'll get a protein crystal. Now, once you get the protein crystal, it's a solid, okay? You can take it and then you shine X-ray on it, right? You blow X-ray on it, and then this X-ray, as it touches the crystal, will have a diffraction pattern. Okay, so this is why you should take physics, okay? You'll learn about how diffraction works. And once you get these diffraction patterns, so it essentially looks like this. Now, no one can see this and immediately tell you what it is, especially nowadays. So then you feed this data into a computer, and this computer, through analysis, will give you a electron density map. Okay? And from this electron density map, you can sort of assign what elements are there. For example, we know that oxygen captures a larger electron density than carbon. So if you see electron density of a particular order, then you know that's probably oxygen, and you know protein has a structure of amino acids. You know how amino acids look like. So then you can use computers to fill in these electron density maps and what they represent. Okay? So from that, you can get your protein structure. Now, out of these three steps, okay, number 2 and number 3, not too hard. Okay? Step two is just shine the X-ray on it. As long as you have an access to high energy X-rays, that's not bad. Step three is computer analysis. That's not bad. You can actually use programs that people have already written and then use it. The hardest step is actually number one, how to get a protein crystal. Okay? Not all proteins will crystallize using the same buffer. Okay? So you'll hear stories about PhD students trying to crystallize their proteins for 3 years, and then they still can't get a good crystal. Right? It's because every protein has a different characteristics, and because every protein responds differently to a different solvent, there are companies selling you these things using a sort of like a high throughput plate. Each wall has a different solvent, so then all you have to do is add your protein in there, and then you wait. Okay, so literally, you can actually go talk to people that do these. Right? So I have a friend who used to crystallize proteins, and essentially what he does every day is he go to the refrigerator and see if his thing precipitated. Right? It's not super exciting, but this work is important. Right? Don't get me wrong, this is pretty important. Now, a lot of times you have no idea like how to crystallize your protein, but once you get a good crystal, then you can do your X-ray crystallography. Okay? All right. So high energy X-ray. Where do you get those? Okay. Typically, people use the synchrotron to get that light source, this high energy beam, which you need to get a good resolution of your particular protein. Now, most of the structures that we get nowadays is usually around 2 angstrom. Right? Now, 2 angstrom is actually pretty good resolution. Okay? You can see where each atoms are. Really close to us, we have Taiwan's National Synchrotron Center, or National Synchrotron Radiation Research Center. It's really just across the street from the back, right? So you can actually walk there. It's not far, about 15 minutes from this building. Okay, you can go there and then request access. Of course, you have to pay the money. Request access to their beam, and then you take your sample, shine a beam on it. You can get the structures. Okay, so it's quite good. Of course, there are larger synchrotrons. The larger the ring, meaning higher the amount of energy that you can get. Higher amount of energy, the better the resolution that you can get. Okay. All right. So once the structure of the protein is determined, you can actually do quite many things. Like I said, you can study the active site of an enzyme. Okay. You can use the structure to design inhibitors for drug, engineering enzyme binding pockets to change substrate specificity, and many others. Now, this is something that I want you guys to remember. Amino acid residues in a protein structure can be viewed many different ways. Now, these are just computer softwares that help us to visualize your protein. In reality, your protein will look more like your surface, so it's like it's a glob, okay? But a glob structure sometimes is not useful for us because sometimes we want to see the inside of the protein, right? So there are many different ways that you represent protein structures. For example, you can have lines, sticks, spheres, surfaces, mesh that looks at electron density, dots, ribbons, and cartoons. Okay, so these are different ways that you can look at your protein structure. Now, there are softwares that I presume maybe you guys have actually learned this. 你们那个大学部的那个计算生物课有没有用过 PyMo 或者是去用那个 看 protein structure？ 还没 ，OK。应该会有个实验课，他会 actually 教你用一些这种东西，这其实蛮好用的。There are many different programs, but essentially, you can grab a structure and then you can, rotate it like you're playing a 3D video game, okay? And then you can see like, oh, where the substrate binds and that kind of stuff. Typically, protein structures, when you see them in a journal or when you see them on a computer software, most people like to use the cartoon figure to represent the protein. Okay, so as you can see here, you have 2 subunits. They form together a functional protein. You see these ribbons and lines, these arrows and ribbons. Okay, we'll talk about them in our next class or later today if we have time. They represent the secondary structures of your protein, the alpha helices and the beta sheets. So Using this, people can do a wide range of analysis of their proteins. The last thing that I want to talk about for this particular lecture is chemical peptide synthesis. Just like when we talked about making DNA, oligonucleotides, making short DNAs for primers, sometimes you can also find uses in making small peptides. Okay, for example, a lot of small peptides are involved in cell-cell signaling, trigger for certain things. Okay, so it becomes useful if you have a chemical method to synthesize oligopeptides that can directly be used for whatever experiments that you're interested. The chemical synthesis for polypeptides is also using solid phase. It's a solid phase peptide synthesis. All right? Now, the basic principles are, number one, it should be possible to add amino acids one at a time, preferably in an automated manner so that people don't have to sit there and add solutions for you. Number two, the amino acids must be activated in some way such that peptide bond formation is both favorable and efficient, so that each cycle you have near 100%. Because if you don't have near 100%, what happens? The same problem in Edman degradation happens. You're going to get a mixed population of your polypeptide. That's not good. And then three, all reactive groups must be protected to avoid side reactions. That's important because you don't want to have an ugly looking protein. Number four, the protecting groups or protection groups used for side chain groups must be stable for the entire synthesis, but the protection group for the alpha amino group must be removed selectively for each cycle of the peptide bond formation. Okay, so for every cycle, the alpha amino group must be able to react, because otherwise you don't have a polypeptide. So in short, it works like this. Again, you have some kind of a resin, a bee somewhere that traps your growing chain. So it's already loaded with one of the first amino acid. Now, what do you notice? You're adding amino acid from which terminus? You're adding it from C to N terminus. Okay, the head is going to be right here, the C terminus. And then the next base that add to it is going to add to the N terminus. Okay, so just like synthesizing DNA, it's going to be in the reverse of the natural process. All right. So what happens is this: your amino acid is going to have a functional group attached to the alpha amino. It serves as a protection almost. And then you're going to activate your amino acid first. Okay, so you're going to activate the acid such that it becomes an activated version of it. And then what happens is this activated version will then be added to the solid phase. Okay, so your beads. And then you have a nucleophilic attack from the amino group of the base of the residue that's already on the bead. You attack it, blah, 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 and then you kick out this particular leaving group, and then you add it on your next amino acid. Okay, so initially you have the first residue, now you have the second residue. This second residue has a protection group on the amino group, okay, by itself. You have to get rid of it in order for this process to repeat. What's the reason of this protection group? Because if you don't have this, then it can react with itself. You need to have this protection group, and then after you remove it, okay, using acid, then you take this guy, comes back up here, right? And then you can start again. What do you notice? Okay, is again the same question from Edman degradation I have for you. How do you stop the reaction for one cycle? Meaning, how come? Okay, how come? Let's say you have bead glycine. The next base you want to add to it is alanine. Okay, so you want glycine, alanine. Okay, so the second base you want is alanine. And the third base you want is serine. Okay, whatever. So you want glycine, alanine, serine. Okay, how come? Okay, how come you don't have glycine, alanine, Okay? How come you don't just add another alanine on top of this alanine? How come you don't continue to react? That's something that I want you to think about looking over this process. Okay? Looking over this process, look at how is it that I can finish one entire phase before I restart and then do the other addition. Okay, I want you guys to think about that. Think about how is it that you can add one base at a time. Okay, how come you don't add, just keep on adding alanines onto the same bead? Okay, think about that. So this is you're adding, and then you're done, right? At the end, you want to cut it off. Okay, you want to liberate your oligopeptide from the bead, and then how are you going to do it? So it turns out you're going to use a strong acid, in this case HF, right? HF, not something very friendly, but it's okay. You add it in, elute it, fluoride is going to be attached to the bead, and then you cut out your acid, and then this thing, since it's liberated, it comes out. Okay? And then you collect that, that is your oligopeptide. Okay? And then you can use your oligopeptide for whatever purpose that you guys are interested in using it for. So, short summary for studying proteins. Right? So we talked about many methods for protein purification in our last class. Now, some of the information that you can or should get for identifying your protein, these are some of the information that you can get. You can get the amino acid composition. You should know how to do that. You can get the PI of the protein. You should know how to do that. You can get the size of the protein. You have a very rough method and a very exact method. Think about how you can do that. You can get a sequence of the protein by using Sanger reagent for getting the first N-terminal amino acid identity, or you can use Edman degradation to get short segment reads, or you can use mass spec coupled with genome sequence database analysis. Okay, then you can get the sequence of your particular protein. You can get the structure of your protein. You should know how to do that. Okay, so overall, these are some of the information that you can get for any protein. And pretty much you can get the biochemical and the structural data that you need to do whatever that is you're doing. The only thing I haven't talked about is protein-protein interaction. Okay, for example, this particular protein, who does it interact with? But that's for molecular biology. Okay, cool.